Hello everybody, welcome. I would like to introduce Dr Claire Wallace, who is our speaker for tonight. Claire?
Oh, yeah, fantastic. Okay. Hi everyone. It's nice to see you all here this evening. So, as Trudy mentioned, my name's Claire Wallace, and I work for the Butterfly Conservation Trust as an ecologist. Before I begin this talk, I would just like to point out that all views and ideas presented today are my own. They're not representative of the Butterfly Conservation Trust. So before I worked for the Trust, I did my PhD, which was looking at the impacts of road verges on bumblebees. And then prior to that, I was looking at the impacts of rewilding on pollinators more broadly. And then even further back, before that, at undergraduate level, I was looking at the impacts of neonicotinoid pesticides on a particular pollination behaviour in bumblebees. And I'll talk about that in a bit more detail later on. But it's just to kind of give you a bit of background as to why I'm interested in neonics, and I have been involved in this for quite some time. So, before I actually begin, I would also just like to say that I will use the words insecticide and pesticide interchangeably throughout this talk, but they both, in the context of this, just mean the same thing. So the layout kind of for the talk today, the first thing we'll talk about is what neonics are and how they work. Secondly, I'll talk about how they impact bees, and this will form the kind of majority of my talk because most of the research on neonicotinoids focuses on bees. Then I will go on to talking about how neonics impact our waterways. This was mentioned as it's a particular interest to your group, so I'll talk about that a little bit as well. I'll then go on to discussing what policies are currently in place regarding neonicotinoids, both in Europe and the UK. And then finally, we'll have a quick chat through the future and what this the future looks like for neonicotinoids here in the UK. But before I do any of that, I would first like to take you back to sort of the late 1930s, and we're going to talk about this chemical here. Some of you may be familiar with it, some of you may not. I am not going to try to pronounce that name at all because I will butcher it, but it is more commonly known as DDT, and I'm imagining that's what most of you will know this chemical as anyway. So DDT is a insecticide that was used throughout the 40s, 50s, and sort of early 60s, and it was first synthesized in 1874, but it was actually recognised as a really good pesticide in 1939. And since its sort of inception and introduction onto the market, there were concerns from conservationists about this pesticide, particularly the impacts it had on birds. So ornithologists were very concerned about how this pesticide impacted birds of prey specifically. The reason for this is because this pesticide does something that we call bioaccumulate. So the insect would come along and obviously ingest the pesticide. Between the time of consumption and time of death, these insects could be eaten by a plethora of other predators, notably kind of small birds. These birds would then ingest the pesticide as well and would then go on to be eaten by birds of prey. And as this pesticide made its way up the trophic levels and through the food chain, it became more concentrated, and that posed problems at the top of the food chain and the birds of prey. And what it did was actually inhibited them from producing calcium. So the eggs of these birds were much thinner and not viable. And so the chicks didn't survive and populations of raptors declined significantly. And this kind of culminated in the publication of this book here. So this is a book by Rachel Carson called Silent Spring that was published in 1962. And it kind of was the catalyst for the inevitable, which was the ban of DDT in the 70s and 80s. So the US banned it in 1972, the UK following suit in 1984. And it's globally banned now worldwide. From 2004, it was recognised across the world. And you might be wondering, well, why am I talking about this? The reason I'm discussing this at the beginning is because when DDT was banned, it left a huge gap in the market. we needed food and we needed pesticides to prevent pests from eating that food. And this was why neonicotinoids were introduced into the market. And I'm going to refer to them throughout this talk as probably neonics, just so that you know that's what I'm talking about. So, bit of history on neonics then. The first neonicotinoid, imidacloprid, was launched by the chemical company Bayer in 1991. And they are a group of pesticides similar in structure to nicotine, hence the name. And there are many, many of them, but for the purposes of this talk, I'm only going to talk about these 5. The reason being is because they are the most commonly used and they are also the ones that are most prevalent in the literature. So these are imidacloprid, which was the first one, clofeniadin, thiamethoxam, thiacloprid, and acetamiprid. So we'll discuss all of these in turn. These are the most widely used group of pesticides across the globe, and they cover more than 25% of all insecticides sold and used. And they have a market value of about $3 billion US dollars. So it's a huge, huge industry here. They're primarily used for sap-feeding insects, so things like aphids, which will bite into the plant itself and then use sucking mouthparts to remove the sap from the plants. And there are different ways that these pesticides can be applied, and the most popular one is via seed coating. So you can actually coat the seeds of the crop in the pesticide before planting, and that saves the farmers a whole lot of work because they don't need to actually apply the pesticides themselves at all. You can also use it as a spray, and I've also included here that neonics are used in pet parasite treatments, and I'm going to speak about that when we come to talking about aquatics, but that's just so that you're aware that they're also used in this form as well. So now that we've got a bit of a brief introduction as to how they came about, what actually are they? So neonicotinoids are neurotoxins. So they target the nervous system of pests. And how they do this is they bind to receptors, enzyme receptors in the nervous system. And this excites the nerves and will lead to negative effects sort of in performance and behaviour of individuals, and subsequently the death of those individuals. These particular pathways are much more common in invertebrates than they are in vertebrates, and so they selectively target insects. And something that is going to become really important as we go through this talk is that bees have much more of these neural pathways than other insects do. Bees are what we would class as an intelligent insect. They're able to learn, they use navigation tools, they're very, very smart. And so they have much more of these pathways than other insects do. Bees also have less detoxification genes, so they're not actually able to flush these pesticides out of their systems as effectively as other insects are, which again has kind of culminated in these negative effects. Another thing that's important to note about neonicotinoids is that they are systemic. They are very persistent. Once they are applied, that's them in the soil. They'll last the entire season. So it's very popular for farmers to just spray them or seed coat them, and then they don't have to do any work. So they're very time efficient, cost efficient. And this also means that these pesticides are incorporated into all of the plant tissues. So not just the stems and the leaves, but also the floral rewards of these plants, so the nectar and the pollen, which is why they've become such a problem when we're looking at bees and other pollinators. Before I go on to talking about the effects, I'd just like to clarify that there are 2 kinds of effects that you can have from pesticides. The first are your lethal effects. So these essentially cause direct mortality and kill the individual. The second are sublethal effects. And these actually reduce the performance of an individual and alter the behaviours so that they're not as efficient as they could be. And throughout this talk, when I talk about effects, I will be talking about the sublethal effects unless stated otherwise, just so you're aware of that as well. So moving on to what the problem is essentially. So as with DDT, there was concerns about neonicotinoids from the start, from conservationists, but rather than being from ornithologists and birds, this time it was from the entomologists. because these are kind of target central nervous systems of insects, they were considered of concern. And these concerns were justified. So some things that started to come up into the kind of woodworks here, and the first sort of literature about neonics. The first thing that they noticed was that neonics were having an effect on the learning capacity of honeybees. So honeybees weren't able to learn behaviours that they otherwise would be able to learn much quicker. So things like the waggle dance, for example, was impaired by the presence of neonicotinoids. It was also discovered that honeybees had problems with flight and navigation after being exposed to these pesticides. So bees use landmarks to recognise where they are and when they're out in the landscape and returning to the nests. And lots of honeybees were actually failing to recognise these signposts and getting lost and not coming back to the nest at all. There was also a reduction in the sensitivity to kind of tastes and smells in honeybees. So they weren't able to detect good quality nectar and pollen from not so good quality nectar and pollen. And you can imagine as they kind of forage and bring things back to the nest, they want the best thing that they possibly can to bring back. And so this was having knock-on consequences for colonies. And the kind of final thing that was noticed was that honeybees were much more susceptible to disease. Originally, when this was presented, people were concerned about neonics' impact on colony collapse disorder, which hopefully some of you will be familiar with as kind of this disease, as it were, that's causing the collapse of these colonies. However, evidence has since come out that neonics are not tied to this, but they are tied to other kind of parasites, so kind of internal parasites like Cryphidia, Nosema, varroa mites and things. Honeybees are much more susceptible to when exposed to neonicotinoid pesticides. And all of these things led to an overall reduced performance in the colony and a reduced fitness of the colony. So they weighed much less, they were producing less honey, they had less workers, less drones, and less new queens were produced. So overall, a negative impact on honeybee colonies. And so this was kind of the start of the problems with neonics. And in response to this, the chemical companies had some critiques of the research that was coming out. And there were four main ones. The first was that these studies were not using field realistic doses of the pesticide. They were exaggerating how strong the concentrations were in the field and applying that to the colonies instead of what you can find in the field. They were also concerned about the long-term exposure. So many of these studies exposed bees for a short period of time to the pesticide rather than for the duration of the cycle as would be experienced in the wild. And you might think, well, surely that's even worse if they're getting exposed to pesticides over and over. Why would the chemical companies bring this up? But the reason for this is because insects often become habituated to stressors. So they can become used to them and adapt their behaviours accordingly back to how they should be responding. And so that was another critique. Another one was that all the studies were lab-based. So none of them were actually out in the field where bees would be exposed to multiple different stressors at once, and these were likely to have kind of synergistic effects on colonies that were not observed in these studies. And then finally, and arguably one of the most important ones, is, well, what about other bees? All this research was focusing on honeybees, and honeybees are a completely different species and genus from solitaries and bumblebees, and so these bees might respond differently. They might not have as severe impacts, and so the chemical companies wanted to kind of know this. And these critiques are all very valid, and I agree with them all, and as did the scientists at the time. And so they responded by creating a whole new realm of research that addressed these particular concerns. And so what I'm going to do now is, instead of kind of surface level going through loads of different studies, because there are thousands of them that investigate the impacts of neonicotinoids on different kinds of bees, I've selected 3 studies that I would like to go into a bit more detail about with you, so that you've got kind of a more comprehensive idea of what the researchers actually were doing and the questions that they were answering. And I've selected these 3 studies, one, because they're ones that interest me personally, and I've used them a lot when I've been writing up literature reviews and for relevant research. Secondly, all 3 of them focus on a different pesticide, one of the big 3. So the first one looks at imidacloprid, the second one looks at clethodim, and the third one looks at thiamethoxam. So the big 3 pesticides are all covered here. And kind of finally, the last one, case study 3, is actually a study that I co-authored and worked on, so I feel like I know quite a bit about it and I'm able to explain it to you quite well. So we'll start then with case study 1. So this is the paper here. It's Whitehorn et al. 2012. This was led by a group of researchers in the UK and was published in 2012. And this addressed kind of 3 of the concerns of the chemical companies. The first was this was a big field study. It wasn't a lab-based study. The second, this looked at Bombus terrestris, the buff-tailed bumblebee, so not honeybees. The buff-tail is the most common bumblebee species we have in the UK. It was a short-term exposure study, so it didn't address that, but something, for whatever reason, I forgot to put it on here. It did look at field-realistic doses in this study as well. So what did these researchers do then? So they had 75 commercially reared bumblebee colonies, so that's this box here. You can buy in bumblebee colonies if you are a researcher or if you are a grower, a farmer, to supplement your pollination of your crops. And so the researchers did just that. They bought in 75 of these colonies and they subjected them to 3 different treatments. So one group of colonies was fed just nectar, no pesticide at all. The second group was fed a lower concentration of the pesticide imidacloprid. And so when I say lower, you're talking 0.7 parts per billion, so absolutely minuscule levels of pesticide there. And then there was another group of colonies that were subjected to a slightly higher level, so it was 1.4 parts per billion. And the types of concentrations you find in the field range from anywhere 0.1 right up to kind of 20 parts per billion. So this was at the lower end of what could be expected in the field. And they then measured different metrics of colony success. And the one I'm going to talk to you about just now is the number of queens that were produced by these colonies, which is arguably the most important metric of bumblebee colony success, because these queens are what go on to found the next generation the next year. So the more queens, the better. Okay, so along the x-axis here, we have the treatment. So on the left-hand side, you have the control. This was no pesticide. The middle, you have the low treatment. So this was the 0.7 parts per billion. And then on the right-hand side, you have the high. So this was the 1.4 parts per billion. And up here on the y-axis, we have the mean number of queens produced. So the number of individuals that were produced by a colony. And hopefully you can very clearly see there is a stark difference in the number of individuals produced by the control and then the 2 pesticide treatments. And what was really interesting about this study was that there actually wasn't a statistical significant difference between the lower concentration and the higher concentration. So it didn't matter past 0.7 parts per billion how much pesticide you fed them. They were already experiencing a severe negative effect. And this was kind of the first time that this was recorded in the science and the literature. And it's one of the kind of key studies that caused policy changes, which we'll talk about later on. So this was kind of the first case study that I wanted to go through. The second one then is this one. It's Rundloff et al. 2015. So this was based in Sweden, in the south of Sweden. And again, it actually addressed all 4 of the concerns that the chemical companies had. So this was a field-based study. It was looking at Osmia bicornis, or the red mason bee, which we also have here in the UK. And this is a solitary bee that kind of nests in cavities. So it'll nest in holes in the walls, in the ground. If you see bee hotels advertised in your garden centre, this is the species that will use these hotels. It was a long-term study, so bees were exposed for the entire duration of their life cycle. And also, again, not mentioned, but it was field-realistic doses that these bees were exposed to. So what they did was they had 16 matched landscapes in southern Sweden. So when I say matched, what I mean is they went out, they found 8 farms that were treated with, or grown crops where the seeds were treated with clethyniodin seed coating. And they basically then went out and found another 8 farms that were similar to these farms in terms of the crops produced, the size, the soil quality, rainfall, these kind of things, but they didn't have the pesticide present, and they did a direct comparison between the two. And what they measured then was they did 2 things. They went out into the fields themselves, did standardised transects and counted the number of native bees that were present, so solitary bees and bumblebees. And then they also put these sort of bee hotels, bee traps they're called, out onto the farms and measured the uptake of these hotels to see how many individuals were actually nesting there. And for the first part of that study, so the measuring of the native bees, this is what they found. So again, left control, right is the coated, and this is the density of wild bees. And again, you can see that the control had much more bees present than the one that had the pesticides in the environment. And the authors actually accredit this to a thing called flower constancy. So bees exhibit this behaviour where if they find a good patch, a floral resource, rather than go and search the landscape over again, they will continually return to this patch of flowers because they know it's a good resource for them. And what this means that each time the bee is returning there, it's ingesting trace levels of this pesticide, and this can accumulate in the system and potentially cause the death of the individual, and that is what they accredit this kind of lower presence of native bees in the seed coating farms. And then the next thing they found, which was even more stark, was the uptake by the bees of the bee hotels. So you can see they found anywhere between 1 and 5 tubes were filled in any farm on the control site, but not a single colony was founded in any of the seed treated sites. So there were no bees nesting on those farms. And the exact reason for this is not known, whether the bees are detecting that this is not a good environment for them. We don't know how they do that, but there's something going on here, and it is the presence of the pesticide that is causing this difference. So again, this was a really kind of stark result presented to the chemical companies addressing the concerns that they had. Okay, and then the final case study then, this is the one that I was involved in. So again, it was led by Whitehorn in 2017, and this was based in the UK. And this study addressed 3 of the concerns. So unfortunately, due to the nature of the study, it was lab-based, but it did look at Bombus terrestris again, the bumblebee. It was a long-term exposure, so bees were exposed for the entire duration of their lifetimes. And also again, forgot to put it in, but field realistic doses. And this particular study was looking at a behaviour that bumblebees exhibit. So rather than look at the overall performance of a colony, it looked at a very specific behaviour. And this behaviour is called buzz pollination or sonication, and perhaps some of you will be familiar with it. I do have a video here, but the sound quality is not good, so you won't be able to hear why it's called. Oh, can anyone hear that? Yeah. Oh, a little bit, okay. So this is why it's called buzz pollination. The bees produce these really strong buzzes while they're pollinating. But you can kind of see what she looks like here. So the reason this particular behaviour was investigated was, 1, because many of our commercially valuable crops are produced by buzz pollination. Tomatoes, aubergines, peppers, blueberries are all produced by buzz pollination. Secondly, honeybees cannot do this. Only bumblebees can perform this behaviour, and so it's really important that we look into sort of how it's impacted by things like pesticides. And the final thing is that this is a very complex motor skill for a bee to refine and learn. And as I mentioned before, neonicotinoids are neurotoxins. They impact the nervous system, and so there was a high likelihood that this was going to be negatively affected by the presence of the pesticide, which is exactly what we found. But just to kind of explain briefly how buzz pollination works, is in most plant species, or non-buzz pollinated plant species, the anthers of the plant, which are the part that produce the pollen, have sort of a lateral slit down the middle, and pollen is allowed to flow quite freely. So if you think of, for example, a lily, if you touch the anthers of a lily, you get covered in pollen. I'm sure everyone's experienced that before. In buzz-pollinated flowers, the anthers are poricidal. So rather than have this slit, they're actually a tube with a pore at the end. And the bee kind of acts like a key in a lock and key scenario. So to get the pollen out, the bee has to be present. And what she does is she'll wrap her body around the anthers and grip them with her mandibles and her legs. And she then decouples her wings from her flight muscles. So when she then buzzes, her wings don't move, but the entire body of the bee vibrates. And the vibrations, which is what produces this buzzing sound and where it gets its name from. And the vibrations pass from the body of the bee onto the anthers, which cause the pollen inside the tubes to bounce around like mad. And it's eventually expelled out of the pore onto the body of the bee, and she can then pack it into her baskets and move on to the next flower. So it's a very complicated behaviour, and these bees know how to do it from the moment they emerge. It's just an innate thing in their minds. But they can learn to refine this behaviour, so they can get better at it over time. And this was exactly what we measured. So we had colonies of commercial bumblebees, and we fed them, again, different levels of pesticides. So control had no pesticide, the lower dosage had 2 parts per billion, and then the higher dosage 10 parts per billion. So again, well within this field realistic range. And these bees were set up to what we called a flight arena, but it was just a big wooden box with a clear top on it. And we set up a little stick with some flowers on it, and the bees were connected to this via tube and allowed to go into the arena and buzz pollinate these flowers. And we set up an audio recorder so we could hear and record the sort of frequency and amplitude of the buzzes. And we also collected the pollen from the legs of the returning bees, so that we could look at how they were doing in terms of pollen collection as well. And as they were going in, we tagged them with little discs. So they had little coloured discs that had numbers on them. So we could actually track the performance of the individuals from their first visit right the way through to their 10th visit. And this is what we did. And this is what we found. So I don't know if you'll be able to see this right at the back, but I'll kind of use my hands to show you. So there are 3 different lines on here representing the 3 treatments. Along the bottom, you have the trial number. So here is the bee's first exposure to the flower, and here is its 10th. And then up here, we basically have the amount of pollen that was collected by the bee. This blue line here that kind of goes up represents the control. So as the bees are becoming more accustomed and experienced to the flower, they're getting better at extracting the pollen, which is exactly what you would expect. This sort of yellow line here, this is the 2 parts per billion. So again, they are improving, but it is at a much reduced rate compared to the control. And then this red line here is the 10 parts per billion. And you can see it's actually decreasing over time. So the bees are getting worse at collecting pollen. And this could be due to a number of different things, but we kind of attributed it to this accumulation of the pesticide, and it's interfering with the nervous system of the individual. And so it's not able to learn how to get the pollen out, and it's just putting all of its energy into extracting it and getting nothing, basically, because it's not manipulating the flower in the correct way. And so this was a really interesting find, because while we're interested in the performance of the colony and how it's doing, we don't tend to think about how these sort of tiny, minuscule behaviours can be impacted in such a drastic way by these pesticides. And this is likely to have knock-on effects for colony performance, but also knock-on effects for us and crop production, because as I mentioned before, this type of pollination is crucial and essential for tomatoes, aubergines, peppers and blueberries. So without this, we wouldn't have any of those crops. So it's really important for us to kind of understand what's going on here. Okay, with all that being said, I'm going to kind of stop talking about pollinators and move on to talking about the aquatics, as this was mentioned as something that was of interest to the group. So something that is really important about neonicotinoids when it comes to water is that they are water soluble. They readily dissolve in water, which means that they could pose a massive threat to aquatic life. Another thing, neonics have been recorded in the surface water of every single country that they are applied in. So they are leaching into the soil, and then into our waterworks, in every country where they are in use. Something to mention here as well is that the kind of concentrations and persistence of neonicotinoids in water is highly variable. So it can range anywhere from lasting 10 days right up until 2 years in the water system. It can also range in concentration from 0.1 parts per billion right up into the parts per million. So we're changing magnitude here. And there's multiple different reasons for this. So it depends on the water itself, the quality of the water, and whether it's flowing or still, the kind of rainfall at the time, how close the crops are to the water. And something else really important to note here is the clarity of the water is very important, because the three main neonicotinoids, so that's your imidacloprid, your clethianidin, and your thiamethoxam, all are degraded by light. So the clearer the water is, the faster the pesticide will degrade in the water. So although they don't impact vertebrates, they do have the potential to impact inverts, which can go on to have negative repercussions. So inverts are things like, in the water, things like mayflies, caddisflies, dragonflies, water beetles, these kind of things. And some things that have been shown to be impacted by neonicotinoids are behaviours of these individuals. So mayflies and caddisflies will sit on the water and be more susceptible to predation. They don't know why, but this happens with exposure to neonics. They also delay their emergence states, so they'll emerge later from their larval form, which can have negative repercussions on the reproduction cycles of these insects. And there was also a study that actually showed that they will just stop feeding entirely. They will not eat after they've been exposed to these pesticides. The exact mechanism is unknown, but they just stop eating, which is quite drastic. Another thing to note, in the UK, there are thresholds that are considered acceptable levels of neonicotinoids in water, and there are two of these. The first is the short-term acute or lethal level, which is 200 parts per billion in the water. And the second is the long-term chronic or sublethal, which is 35 parts per billion. So with all that in mind, I'd like to talk about a report briefly that was published by BugLife, which is an invert charity here in the UK. Some of you will probably be familiar with this. And they run a scheme called the Water Framework Directive Watchlist Monitoring Scheme. It's quite a mouthful. But they basically go out and monitor our water because they're interested in how healthy it is for our aquatic invertebrates. And they did this in 2016, was the last time it was done. And they surveyed 26 different sites. This included lakes, lochs, rivers, streams, smaller canals. And they found that 74% of these sites were contaminated with neonicotinoids. And this is in 2016, so bearing in mind there was a sort of ban put in place in 2013. So that kind of shows how persistent these pesticides can be. So 74% contaminated. And something that was even more sort of extreme was that 8 of these sites exceeded the chronic levels, so the sublethal levels, so more than 35 parts per billion. And 2 of them actually exceeded the acute levels, one of which was found at 1,030 parts per billion. And the recommended lethal level is 200 parts per billion. So this is a massive, massive increase, which is quite kind of scary and worrying. But this is kind of the most sort of up-to-date, comprehensive guide that's been published in the UK on this. As I said before, most of the literature focuses on bees and pollinators. So let's talk about the policy briefly then, and the response from the governments to the concerns surrounding neonics. And we'll start with talking about Europe. I'm not going to cover the States just because it's quite a bit away, and all 50 states have different legislation. It's just way too much to cover. So we'll talk about Europe and the UK here. So as I'm sure a lot of you will be aware, the EU put a moratorium, so a temporary ban, on 3 of the main neonics in 2013, and this stopped outdoor use of the pesticides. After this was kind of done, they put a call out to researchers and other sort of smaller governmental bodies that they wanted more research done on these pesticides to kind of find out whether they should continue this ban, make it more severe, or get rid of it completely. And after this, what they did was in 2019, based on all the evidence, they banned these pesticides completely, with the exception of emergency use, particularly for sugar beet growers, so they could apply to use these pesticides in emergency circumstances. One of the others, I mentioned 5 at the beginning, acetamiprid, the kind of data on that is very inconclusive, and so it continues to be used and will be continued for use until 2033, is when it's been approved. So we've got another 10 years of that in Europe yet before it's reassessed. And I should also mention, I didn't put it on here, but thiacloprid was also banned, but it was in 2020, so a year later. And then something kind of stark happened this year. On January 19th, the EU announced that they would no longer allow emergency use of these pesticides at all, even for sugar beet growers. They were banned completely for use in the EU, which was great for all these conservationists and for our bees and pollinators as well. They're no longer allowed to be used in the EU. So the UK is obviously slightly different. We left the EU in 2020, which meant that the kind of legislation surrounding our pesticides was up for debate. What was going to happen now that we were no longer kind of subject to what the EU wanted us to do? And a lot of scientists, well, there was kind of a mix of feelings of, oh, well, are we, this is great, it means we can decide to ban them completely outright, and, we don't have to wait on the EU deciding for us. And then there was the other group, which were like, the government's just going to continue the use of these pesticides. And unfortunately, the second group were correct. So after we left, emergency use was applied for in 2021, 2022, and 2023, and all 3 times it was approved for use. And the really sad part is that in 2023, this was just 4 days after the EU announced that the evidence for these pesticides causing harm was overwhelming, and we would not allow the emergency use in the EU. But here in the UK, we allowed it, which is a bit of a shame. But what does that mean then for sort of the future of neonics here in the UK? Well, the reality is it's very uncertain. We just don't know. There still remains sort of a ban on the chemicals. However, there are these emergency uses that are being allowed. And the government has kind of put forward this idea that they want to have an emphasis on integrated pest management for farmers. So rather than kind of going straight to pesticides, we're going to go through all these steps first of, making sure that you're managing the land correctly to reduce pest numbers, bringing in biological controls, these kind of things. But sadly, having worked with farmers and, having farmers in the family, I know that farmers most of the time think this is a complete waste of time, because the inevitability is we're going to have to use these pesticides at the end of the day. Why are we going through all these sort of loopholes to get to that inevitable point? And something that was interesting was kind of mentioned when the government put forward their statement as to why they were allowing the emergency use of the pesticide this year, was this quote. And they said that until there are basically adequate replacements for neonicotinoids, we will continue to use them. We have to, because otherwise our crops are going to suffer and we're going to have lower yields than we would like. And so the likelihood is that the use of these pesticides is probably going to continue for another 5-10 years in this country until we are able to kind of get that sort of push to have them banned like they are in the EU. But that's kind of a very sort of sad note to end on. So actually what I'd like to do is end on this note. So there are lots of wildlife charities, organisations, groups like yourselves that are out there kind of fighting the good fight and signing petitions and protesting and writing to their local MPs about these pesticides and trying to kind of stop their use in the UK. And so hope is not lost, and I think that they will eventually be banned. They have to be. It's just how long is it going to take to get there? And so I'll end on this quote, which is the kind of slogan from the Bumblebee Conservation Trust, which is, working together, we can save the sound of summer. So I'd like to thank you all for your time and your attentive ears, and if anyone has any questions, I will be more than happy to answer them for you now.